All right, welcome to Nervous Tissue Part 2. We're picking up right where we left off with this reminder of how the material is laid out. My PowerPoints start with the synapse, so where this cell here is talking to that cell there. That communication is what is going to cause the action potential, which will start here and travel down the axon and cause the release of neurotransmitter. The way the OpenStax book goes over it is it starts here with the action potential, and the action potential travels down, neurotransmitter gets released, and then they talk about synapses. I like to start with synapses. You'll see what's coming up soon, is a brief introduction to the action potential so that we sort of know where we're trying to get to. So we want to know what needs to happen here while we're talking about what's happening here. So I do a brief intro and then we backtrack and run the whole way through. It's going to be awesome. So we start just by talking about synapses. This is anywhere where a neuron is communicating with another cell. So it could be a neuron talking to another neuron, or it could be our neuromuscular junction, which is a specialized form of synapse where the neuron is communicating with a muscle cell. There is some terminology that we need to become familiar with. Some of these terms we know already, like synaptic cleft is just the space in between the two cells in a synapse. But now we need to get this presynaptic neuron, postsynaptic neuron terminology down. And the way you want to think about this is in terms of information flow, right? So there's an action potential that's going to be traveling down this axon, and then whatever signal this is, whatever information is being communicated, it's being communicated from this cell to this cell. So the communicating cell is called the presynaptic neuron, and then the cell that is being stimulated or inhibited with, sort of the next cell in the chain, is then the postsynaptic neuron. And when we talk about synapses, and when we get into how neurons work, we're going to start with what's going on in the postsynaptic neuron, so the information receiving neuron. Before we jump into the meat of what we're going to cover, we want to understand different kinds of synapses. There are two different kinds of synapses. One is called an electrical synapse. This is really the last time you're going to hear about it, but it's just a synapse where the presynaptic neuron and the postsynaptic neuron are joined together by gap junctions. So there really isn't much of a synaptic cleft. This is just an image of gap junctions I stole from somewhere because our book doesn't have a good picture of an electrical synapse. So these are channels in one membrane that butt up against channels in the other membrane, forming pores, so that instead of dumping neurotransmitter onto the postsynaptic neuron to open ion channels and let ions flow through, ions can flow directly from the presynaptic neuron into the postsynaptic neuron. So you're skipping the chemical middleman and just having electrical activity in one cell directly affecting the electrical activity in another cell. And again, that's probably the last time you'll hear about this type of synapse this semester. Most often when you're talking about synapses, you are talking about chemical synapses that rely on neurotransmitters and neurotransmitter receptors. We are going to have two different types of chemical synapses with different kinds of receptors. So it's really two different types of receptors that you find at chemical synapses, metabotropic and ionotropic. So when you see that suffix tropic, tropic, that means stimulating or changing. So in a metabotropic receptor is going to alter the metabolism or the physiology of the cell. An ionotropic receptor is only going to affect ion flow. So we will start with metabotropic synapses first. They rely on our good friend, the G protein coupled receptor. So in this case, you have a receptor embedded in the membrane that communicates with this G protein signaling machinery. When neurotransmitter hits it on the outside, stuff happens on the inside. We're not going to go into much detail about G protein coupled receptors, but you just want to know that a lot of different things can happen on the inside depending on what kind of machinery is hooked up to this receptor. So you kind of want to think of the receptor as like one of those Dremel tools, or, oh, I forget the other one. But it's like one of those multi-tools where you can put a grinding bit on it, or a buffing bit, or a cutting wheel, or a sanding wheel. So this would be sort of like the power unit of your rotozip or Dremel tool, and then all different sorts of machinery can be hooked up to it inside the cell. So in the case of metabotropic synapses, this can lead to changes, long-term changes in the physiology of the receiving or postsynaptic cell. These responses, as it says here, are slow, complex, often prolonged. If you're changing the metabolism of a cell, the way it works, maybe even the way it responds to neurotransmitter in the future, you're talking about changing the way that cell works. That's learning and memory. So if you're gonna learn something, you need to change something about a neural pathway to reinforce it, and quite often it's the metabotropic synapses that are altering the sensitivity of cells in that circuit to make that circuit stronger. Your metabotropic receptors can ultimately activate, open, and close ion channels, so you can get sort of traditional depolarization, hyperpolarization results from a metabotropic receptor, but they also can affect more long-term physiological functions of the neuron. Then we get to ionotropic synapses. This is what most people are referring to, or what we usually think of when we say a synapse. This is the kind of synapse we're going to be spending our time talking about, and this is the kind of synapse that is going to mediate most of the effects we're going to see. So just like the neuromuscular junction, that's an ionotropic synapse. Most of the neuron-neuron communications we're going to see in this course are run through ionotropic synapses or ionotropic receptors. So if you have an ionotropic receptor or synapse, this means you have ligand-gated ion channels as receptors, just like the acetylcholine receptor in the neuromuscular junction, so that when the neurotransmitter binds to the receptor pops open. Then, depending on which ion that receptor is for, you might get ions flowing in or ions flowing out. That is going to alter the membrane potential. So now we want to know how these ionotropic synapses mediate communication between two neurons and how neurons function in general. And this part here, I think, is why the book starts with action potential, because you do want to know what the point of all this stuff is, right? Why are you doing all of this stuff at the dendrites? It's so that you can generate an action potential. So what you need to know for now is that the entire neuron at rest has a resting membrane potential of negative 70 millivolts. There is machinery in the axon hillock, voltage-gated channels, which we will talk about later, that respond to changes in the resting membrane potential. When the membrane potential at the axon hillock, where the action potential is going to start, when that is depolarized from negative 70 to negative 55, that is what triggers an action potential. So if you look up here, you've got something happens here. That's what we're going to talk about first, to get the axon hillock here depolarized to negative 55. And you see there's like a little blip there. Then once it hits negative 55, you get a rapid depolarization, repolarization, hyperpolarization, and return to resting membrane potential. So this part here, all of this is the action potential, which we'll talk about later. What we're going to talk about now is how you get from rest up to threshold to generate your action potential. But keeping in mind that the name of the game here is to get your axon hillock right there to negative 55 millivolts, and that's what activates this cell. Let me go back real quick, sorry. You will recall, right, negative 70 is the resting membrane potential. There is almost always some sort of potential across the membrane. So we just refer to the electrical state of the membrane as the something potential. At any point in time, there's a potential, an action potential, an end plate potential, a resting potential. So they're all potentials, and they get named for whatever state the membrane is in at that point in time. So now we are talking about, let me go back again. We'll leave it. What is happening in the postsynaptic neuron? So this is the neuron that is having neurotransmitter dumped on it. And so what happens is the neurotransmitter is going to bind to the receptor. The receptors are ion channels, so those channels are going to open. Ions are going to flow maybe into, maybe out of the cell. It depends on the ion. That is going to alter the membrane potential. So these deflections, or whatever you want to call it, deviations from the resting membrane potential, are called postsynaptic potentials because they happen in the postsynaptic neuron. There are two different kinds of postsynaptic potentials. First are excitatory postsynaptic potentials, or EPSPs. Recall that threshold is negative 55 millivolts, which I don't know why that's negative 15 there. We'll put 55 here, even though the scale is off, right? If this is threshold and this is where you're generating an action potential, which is exciting the postsynaptic neuron, then this action potential is getting you closer to threshold. So it is exciting the cell, but not getting it excited enough to generate an action potential. So EPSPs depolarize the membrane. Then you can get what is called an inhibitory postsynaptic potential, or IPSPs. So in this case, whatever ion channel is opening, it is causing a hyperpolarization of the membrane. So now the inside of the membrane is becoming even more negative. This now means you are farther away from threshold than you were before. This is again referred to as an inhibitory postsynaptic potential. Both of these PSPs, the EPSP and the IPSP, are referred to as graded potentials. They're called graded because they're not all the same, just like an A isn't the same as a B. And they have three important properties. Two are illustrated here. They vary with stimulus strength, or they come in different sizes. Also they can be having trouble delivering lecture today, right? They can be inhibitory, they can be excitatory, and then also inhibitory can be mildly inhibitory or more inhibitory, or you might have, right, an EPSP that looks like that. So they just come in different shapes and sizes and different magnitudes, and diminishing quickly over time simply means that as time progresses, you return to your resting membrane potential. Decreasing in magnitude with distance requires a slightly different graph, or a different way of looking at it. I did just add this 0 right here, so if you don't have it on your graph, that's fine. So here what you want to imagine is that we've placed electrodes, little measuring devices, at these three places. We are going to dump some neurotransmitter onto the postsynaptic cell, and instead of measuring what's happening over time, which is what the graph originally said, I also changed that, we're measuring the membrane potential at the same time, but at varying distances from the axon terminal. So if you're right underneath it, you're 0 units away from the axon terminal, and then the further you go in either direction, you're further away from the axon terminal. So if you measure the membrane potential right after some neurotransmitter gets dumped on this postsynaptic neuron, the effect of that neurotransmitter is going to be greatest right in the middle of the synapse. And then the further away you get from the middle of the synapse, as you get over here, the effect of the postsynaptic potential is less further away. And this is because it is relying upon ions flowing into the cell. So the ions all flow in here. So obviously you're going to measure a lot of depolarization here. Any depolarization that happens over here happens because ions diffuse in that direction. So because depolarization or altering of the membrane potential adjacent to the synapse requires diffusion, right? The effect is only going to go so far, and then it's going to diminish the further you get away. And later, when we look at action potentials, you'll see how they're different and why we organize the information this way. So those are our postsynaptic potentials. Then we need to understand what summation is. So earlier we were talking about, I'm not going to go back to the original slide, I'm just going to skip ahead one, how there's integration in the soma. This is the integration part. It's called summation. So as it says right here, the activities of all of your postsynaptic potentials are summated by the soma. We'll go over how that happens, but basically you have always more than one synapse on any given neuron, and they all affect to different degrees what the voltage at the axon hillock is, and they have sort of a collective effect on the axon hillock. That collective effect is called summation. When you get to -55, you have an action potential. So first up is temporal summation. So the way temporal summation works is that you've got one synapse, so you're looking at the activity at one synapse having an additive effect on the resting membrane potential over a short period of time. So let's imagine we're looking at this synapse right here. This little axon terminal right there is green, so we are going to assume that this is an excitatory neuron, so we're going to be generating excitatory postsynaptic potentials. So you're here with your resting membrane potential. You release a little bit of neurotransmitter, it looks like that. If you wait long enough, you reestablish your resting membrane potential, you release a little bit more neurotransmitter, it looks like that. If you do 2 bouts closer together in time, so you release a little bit of neurotransmitter, and before you get back down to rest, you release a little bit more neurotransmitter, right? Now you have this additive effect here, where if you stimulate this synapse right here, if you dump 2 droplets of neurotransmitter onto your postsynaptic cell in rapid succession, one right after the other, they have a greater effect on membrane potential than if you had allowed time to pass. So this is called temporal summation. One synapse with multiple bouts of activity over a short period of time. With spatial summation, now what is going to be constant is time, so you're going to have activity at multiple synapses happening at the same time. Only in this case, right, if I go back here, right, here it only works if the multiple bouts of activity come with very little time in between them. Here you need to have very little space in between them. So let's then think about, I don't know, this synapse here and that synapse there, but we're going to measure right here. So we're going to have a part of the membrane that could be affected by both of these synapses, and we're going to look at what happens there. So if you just get, and we'll call this 1 and we'll call this 2. Right, so maybe if just 1 fires, you might get something that looks like this. And if just 2 fires, you might get something that looks like that. But if they both fire at the same time, that's a bit of an exaggeration, but those 2 EPSPs stack on top of each other. So 2 synapses close together firing at the same time will summate. So that's spatial summation, close together in time, close together in space, same time. Temporal summation, close together in time, same spot. And this can also happen sort of in the opposite way. If I just go with the eraser here and erase all this. Now let's imagine that we're looking at this synapse right here, which is going to cause EPSPs, and this red one right here, which is going to cause IPSPs. And maybe we're measuring right here, like equidistant between those 2 synapses. So again, this is going to be spatial summation. They're both going to fire at the same time. And we're going to track the effect of both of them firing at the same time right there. So let's say if just, and we'll call this A and B. Let's say that just A fires, it might look like this. And then if just B fires, it might look like that. And I did a small, let me fill that in, a small IPSP for B. Now if they both fire at the same time, because they're close together in space, you might get something that looks like that. So it's this larger EPSP minus that IPSP. And you get just a smaller EPSP. So with spatial summation, excitatory synapses or EPSPs can be counteracted by IPSPs. And this is what's going on in all of your neurons. You've got multiple synapses on the dendrites and the axons of any given neuron, and any given neuron is receiving some excitatory input and some inhibitory input. So you've always got spatial summation and temporal summation happening, EPSPs having additive effects and IPSPs canceling those out. So if you look here, it's like EPSP, EPSP, IPSP, and then nothing, EPSP, IPSP, and then a big enough excitatory postsynaptic potential to get you to threshold, and then you generate an action potential. So it's sort of like, what's the word I'm looking for? The neuron is like constantly running elections with yes votes and no votes. And if at any given point in time there's more yes votes than there are no votes, or if you get enough yes votes, then ba-bam, you generate an action potential.